welcome you all to my video on pyrosequencing. It is one of the modern DNA sequencing or next generation sequencing methods to find out the order of bases. In 1993, it was first developed using solid phase sequencing with streptavidin coated beads. Then, this technique was modified in 1998 by Ronagi, Erlin, and Niren. Swedish company named Pyrosequencing marketed it and commercialized it. Pyrosequencing is based on sequencing by synthesis principle. Like Sanger sequencing, it also involves DNA synthesis, but this technique depends on many enzymatic reactions in which pyrophosphate is released on the addition of a nucleotide and light is emitted. The light production is very important event here because that's how the addition of a nucleotide is detected. The term pyro has been derived from a Greek word pyre, which means fire. Since here the light is generated, hence the method is called pyrosequencing. The pyrosequencing can be of two types, solid phase and liquid phase. In solid phase sequencing, the DNA fragment to be sequenced is immobilized on the solid support. That means here the solid support is needed and immobilization is also done. However, in liquid phase sequencing, no solid support is needed, no immobilization is needed. The second difference is solid phase sequencing is a system where three enzymes are required. These are named as DNA polymerase, ATP sulfurylase, and luciferase. Whereas in liquid phase, besides these three enzymes, an additional enzyme known as a pyrase is needed. Hence, this is 4-enzyme system and 4 enzymes are needed to complete the process. In the third one, after each step, there are many unused nucleotides left, and these excess nucleotides have to be removed by washing. Then only the next nucleotide can be added. But in liquid phase, this washing step is not required. Instead, there is a pyrase enzyme which is added, which degrades the extra nucleotide. And the last difference is that solid phase is more tedious as compared to the liquid phase sequencing because immobilization and washing steps are required in solid phase sequencing. These days, however, liquid phase sequencing is a preferred technique. Pyrosequencing is the real-time sequence determination technique. It employs 4 enzymatic reactions in a single tube in liquid phase sequencing. We'll discuss all the reactions one by one. In first reaction, pyrophosphate is formed which contains 2 phosphorus atoms. Here you can see the DNA polymerization reaction. N number of nucleotides have already been added to the DNA chain. Now one by one a nucleotide or DNTP is added and the chain is thus elongated by one nucleotide. So now you can see N plus 1. The enzyme which catalyzes this reaction is known as DNA polymerase. As a result, a pyrophosphate molecule is liberated, which is used as an indicator. Remember this thing here, if the nucleotide is incorporated, then only the pyrophosphate is released. Otherwise, no pyrophosphate is formed. And the pyrophosphate formed is proportional to the incorporated nucleotide. That means if one nucleotide is added, one pyrophosphate will be formed. If two nucleotides are added, then 2 molecules of pyrophosphates will be formed. The second enzymatic reaction is about the ATP formation. Pyrophosphate, which has already been formed, triggers this reaction and the APS or adenosine phosphosulfate is used as a substrate here. The enzyme which catalyzes this reaction is ATP sulfurylase, and the quantitative conversion of pyrophosphate to ATP takes place. So that means ATP is the product of the second reaction. This figure shows that the ATP thus formed in the last reaction acts upon a substrate known as luciferin in presence of luciferase. ATP senses the enzyme luciferase. This is the same enzyme which is found in firefly in nature. Oxyluciferin, AMP, pyrophosphate and carbon dioxide are formed during the reaction. And on the top of everything, light is generated. The intensity of light produced is directly proportional to the number of nucleotides incorporated. If 2 cytosins are added, then intensity will be more as compared to 1. The light is detected by a photon detection device or CCD camera. A pyrase enzyme is the one which is used for degradation of unused NTPs. Whenever dNTP are added in the tube to perform the DNA synthesis, one or a few nucleotides are added and the rest are left unused or unincorporated in the solution. It is essential to remove these unused nucleotides. In solid phase sequencing, it is done by washing after each step, but in liquid phase, this function is performed by a pyrase enzyme. Now let us discuss procedure or mechanism. The DNA to be sequenced is broken up into fragments of around 100 base pair. Then the double-stranded DNA is converted into single-stranded by unwinding it, which is called as denaturation. The easiest way to do the denaturation is by heating. An adapter is attached to single-stranded DNA. This adapter is a double-stranded or single-stranded molecule, and like primer, it can be chemically synthesized. In solid phase sequencing, a biotin bead and a magnetic bead streptavidin are also attached to it. It helps to immobilize the DNA to solid surface. To start the DNA synthesis, a primer is required which can be synthesized as per need. This is complementary to the adapter, so it can be annealed easily when these two are incubated together. Once the primer is annealed, the DNA synthesis will start at 3' end, which has a free OH group. If you have seen my video lecture on Sanger sequencing, you will understand the synthesis process easily because in that lecture I have shown it with many diagrams. I'm giving the link here. Since large amount of DNA samples are required, so the PCR can be run to amplify the DNA. This single-stranded DNA will act as template for DNA synthesis. Here I am showing you one of the sequences. Suppose it is ATGT, ACC, GT, AAA, AAA, TC. So if this is the sequence in the template, then now we'll see how the nucleotides are added in the newly synthesized strand and how these nucleotides are detected during the synthesis. Now we'll discuss how the nucleotides are detected during the synthesis. The enzyme which is required for the DNA synthesis is DNA polymerase. As you all must be knowing that other prerequisites of DNA synthesis are all 4 types of nucleotides that make up DNA. For example, D-CTP, D-GTP, D-TTP and D-ATP, which we add one at a time during the process of chain elongation. But there is a complication here. I have explained earlier that ATP is released during one of the enzymatic reactions. And here we also add DATP as a nucleotide. Therefore, to avoid the confusion between the ATP released during the process and the DATP provided from the outside by us. What we do? We add DATP alpha S, which is known as deoxyadenosine alpha thiotriphosphate. That is used here. This is an analog of normal DATP. DNA polymerase ignores the difference and uses it, but luciferase does not recognize it, so the false signal of light is avoided. Okay, so up to this point, I told you about the DNA synthesis. Let us now talk about the other reactions which are necessary to detect the addition of a particular nucleotide through light signal and how this exactly is done. That means the mechanism. The nucleotides are added to the valves or tube which begin to be incorporated onto single-stranded DNA template by polymerase enzyme at 3' end of primer. Here it is to be noted that only one nucleotide will be added at one time. As in this figure, DCTP is added. Here you can see that the C was added here, which is complementary to G. And we always know which nucleotide we have actually added. The addition of a nucleotide like CTP results into the release of a pyrophosphate. Now the question is, where does it come from? You know, the nucleotide is a triphosphate molecule, means the nucleotide has a triphosphate, and out of these 3 phosphates, one phosphate forms the bond along with the OH group of which is present at the 3' end of another nucleotide, and the rest 2 phosphates will be released in the form of pyrophosphate. And remember that this happens every time the reaction takes place or the nucleotide is added to the polynucleotide sequence. In next step, ATP sulfurylase reacts with this pyrophosphate and converts it into ATP. Of course, the substrate of this reaction, APS, is present here, although we are not showing it here in this figure. In the next reaction, ATP converts luciferin to oxyluciferin in presence of an enzyme, luciferase, and the light emission takes place, which is picked up by detector. Here also, the byproducts of the reactions like AMP, pyrophosphate, carbon dioxide are not shown here. Now, let us see what happens to the rest of the nucleotides which are left there unused. Whatever method you use, but these have to be removed. In solid phase, washing is done after each step, and in liquid phase, an additional enzyme is used named as apyrase. It degrades all the unused, unincorporated DNTPs and clears the reaction mixture for the addition of next nucleotide. So now the next one is added, means the next nucleotide is added, and the same reaction will take place and light will be generated. Similarly, we'll keep on adding ATGC nucleotides sequentially. Now, imagine a situation where wrong DNTP was added. Wrong DNTP means the one which was not complementary to the template base. In this case, no addition of nucleotide will be there, so no light will be seen. To understand it, look at the template strand and the newly synthesized strand in the figure. After incorporation of C, the next nucleotide should be A, because in template there is T present. But what we did, we added G or we added T and C, anything other than A. We didn't add A. So naturally these G, T, C, they will not attach in the chain. If they are not attached to the chain, so no pyrophosphate will be released, no further reaction will take place. therefore no light will be emitted. So that's how we'll come to know that the new nucleotide which was added was the wrong one, and we'll keep on adding the nucleotides until the light is generated. One more thing I would like to mention is that the intensity of light is also important. We have discussed this in one of the previous steps also. If two same type of bases are consecutively added, intensity of light is going to be more. In our sample or template sequence, two C's were there, cytosine bases were there, and three A's, adenosine, were there. So we'll be able to detect this also by the peaks of signal in pyrogram. After the whole reaction is complete, we get the pyrogram, which shows nucleotide added at x-axis and intensity of light or signal at y-axis. First nucleotide added was T, and here you can see a peak. Second one was A, and again the same size peak was formed. But after that you see there is no peak. No peak is the indication of addition of wrong nucleotide. Wrong nucleotide means not complementary to the template. Then after TACAT, you see a bigger peak. This indicates the addition of two similar nucleotides. Here, in this case, there were two G's which were added. And there is one highest peak also seen, which shows incorporation of T 3 times, TTT. So that is how we can interpret the sequence or base order of template DNA or say sample DNA, which in this case is A, T, G, T, A, C, C, G, T, A, A, T, C. So that is all about the procedure of the pyrosequencing. Pyrosequencing has many advantages also. A large number of samples can be analyzed in short time. Then it has it has high accuracy, flexibility, and it is very rapid also. It just takes 2 to 4 seconds from the point where the addition of nucleotide was done to the point where the light is generated. And here no radioactive labeling is required. Even the gel electrophoresis is also not required. So this is quite good method, and these days this method is used very frequently. So that's all about the pyrosequencing. Thank you very much, students. And if you like my video, then please go through my other videos and please share it and subscribe it. If you have any questions, you can write in the comment section. Thank you.